Hello viewers, in today's lecture I am going to discuss excretion in Annelida. But before starting the discussion, I want to mention that this is an initiative of Zoological Society of Assam. The rationale of the discussion is to explore the methods of removal of unwanted substances in Annelida. The objectives to describe the various types of excretory organs found in Annelida and to expose their functions and to trace the path of excretion in Annelida. The definition of excretion. Every living organism generates waste in its body and has a mechanism to expel it. Excretion is the process of removal of toxic waste from the body of an organism. It is an essential process in all forms of life. It is a physiological process and is strictly found only in animals. And why it is so much essential? Every organism must rid itself of potentially harmful byproducts of its own vital activities. This process in living things is called elimination. Through excretion, organisms control osmotic pressure, the balance between inorganic ions and water, and maintains acid-base balance. This process thus promotes hemostasis, the constancy of the organism's internal environment. The excretory substances of Annelida. The waste products and the nitrogenous byproducts of metabolism are called excretory substances. The excreted material or substance may be called as ejecta. The major excretory products of annelids includes ammonia, urea, amino acids, water, and different types of ions. Excretory organs of Annelida. The organs of excretion make up the excretory system. Every animals have certain and specific organs that are responsible for elimination of waste from the body. These organs are called excretory organs. Excretory organs of Annelida are categorized into two types: the primary excretory organs and secondary excretory organs. The primary excretory organs in Annelida are the Nephridia, while the secondary excretory organs of these animals include COELOMODUCTS and Chloragogen cells. About these three organs we will discuss in the next slide. COELOMODUCTS acts as secondary excretory organ. These are very primitive and mesodermal in origin. These are developed from the gonads. They grow out and open to the exterior by a genital pore. The coelomoducts primarily function as gonadoct. They carry the gametes to the outside, to the exterior, and along with the gametes, they also expel some sorts of excretory products. Therefore, they act as secondary excretory organ in these organisms. The Nephridia. The ectodermally derived Nephridia are the primary excretory organs of Annelida. These are segmental organs arranged in pairs. A pair of Nephridia is present on each segment. They are ectodermal in origin. They develop from external surfaces, grows into the body, opens to the exterior by a nephridiopore. A typical nephridium consists of a nephrostome, a long tubule or nephridioduct, and a bladder with a nephridiopore. The nephrostome is a funnel-like component of a nephridium, especially metanephridium. It is always oriented towards the coelom. The nephrostome is covered from inside with cilia, which push the water, metabolic waste, and other substances into the metanephridium. The nephridia are lined with short hair-like structures called cilia. These cilia beads repeatedly, which drive fluids down the nephridioduct and thereby creating a lowered pressure within the tubuli lumen. This lowered pressure draws body fluids carrying waste across the thin cell membranes and into the duct. The functions of nephridia. As we know that the nephridia are porous, long, thin and coiled tube. The primary function of nephridia is excretion. However, they also serve to convey the gametes to the exterior. Besides these organs, also maintain osmoregulation. Let us have a look into a diagram which shows the positions of the nephridia or the excretory organ in pheretima. The pheretima, postuma, nephridia consist of a nephrostome which leads into a long coiled tube called the collecting tubule, which enlarges at its posterior end to form a bladder, and this bladder finally opens to the exterior through a nephridiopore. This diagram shows the two segmental positions of the nephridia or excretory organs. The types of nephridia. Nephridia are categorized into several types based on certain features. Based on the tubule, whether the tubule is closed or open, nephridia are divided into two types: Protonephridia and Metanephridia. The Protonephridia are very primitive types of nephridia. These nephridia terminates in the coelom as blind tube. A typical Protonephridia terminates with specialized excretory flame cells or solenocytes. Protonephridia are found in Phyllodosi, Vanadis, etc. In comparison, the Metanephridia are more advanced type of nephridia. Internally, they open into coelom by a ciliated funnel called nephrostome. Metanephridia do not bear solenocytes. Metanephridia are found in lumbricus, leech, etc. About both of these, the Protonephridia and Metanephridia, will discuss later on in details. Protonephridia. These are considered as the most primitive forms of nephridia. Basically, these are the excretory organs of larvae and other adult polychaetes. They have a tubular arrangement that opens outside the body by a nephridiopore. They have tubes with blindness where cilia-mediated filtration occurs. They have terminal or cap cells which can be mono or multiciliated that supports infiltration process. Protonephridia are divided into two types based on their terminations. These are phlegm cells or keratocytes and solenocytes. The phlegm cells. The protonephridia with blind ends that terminates in phlegm cells. These cells are bulb-like in appearance, hence also called phlegm bulbs. These cells bear a tuft of cilia within a cup-like projection whose beating is like a candle flame. A phlegm cell is a specialized cell. It consists of the cytoplasmic processes, ciliary phlegm, an intracellular duct with certain secretory materials. The intracellular duct joins with the excretory duct. The second type of protonephridia are the solenocytes. Each nephridium bears about 500 solenocytes. These are flagellated, tiny, rounded nucleated cell body and a long hollow stalk or tubule. Solenocytes can be distinguished from phlegm cells as the former is usually flagellated, whereas the latter is ciliated. The solenocytes bears a flagella, whereas the phlegm cells bears a tuft of cilia. These organelles, the solenocytes, are associated with excretion, osmoregulation, and ionoregulation in many annelids. These are found in phyllodocytes, alciopedes, and a few other members of. The figure shows the protonephridium of phyllodocy. The protonephridia is associated with the coelomoduct funnel, coelomoduct, and nephridia-duct. The second figure shows more closer view of protonephridia with protonephridial tubuli. The third figure shows in detail view of solenocytes, which consist of a nucleus and flagellum. Metanephridia. These are far advanced nephridia found in vast majority of annelids. It is a nephridium that originates in a ciliated coelomic funnel. In metanephridium, the inner end of the tubule is opened and remains in the form of a nephrostome. A typical metanephridium consists of a nephrostome, a short canal, and a nephriduct, and opens through a nephridiopore to the exterior. Metanephridia are found in oligochaetes, polychaetes, leeches, etc. Let us have a look into the metanephridium of an earthworm. It consists of a nephrostome which leads into a long coiled tubule. The posterior part of the tubule broadens into a bladder which opens to the exterior through a nephridiopore. The second category of nephridia are based on size and number of nephridia per segment. Based on size and number, they are categorized into micronephridia or meronephridia and meganephridia or holonephridia. The micronephridia, as their name suggests, they are smaller in size. and numerous per segment. The networks of fine tubes lying on the body wall and septa in each segment. The Nephridia of Pheretima postuma are micronephridia types. Whereas the meganephridia, as its name suggests, larger in size, but generally only one pair per segment. Usually they extend over two segments and their nephrostomes open into the next segment in front. The Nephridia of Hirudinea are the examples of meganephridia. The third category of nephridia are based on site of opening. Based on the site of opening of the nephridiophore, the nephridia are classified into two types, exonephridia or ectonephridia and enteronephridia. The exonephridia directly opens to the exterior. Nephridiophores are present in case of exonephridia. The integumentary micronephridia of Pheretima postuma, meganephridia of Neris, Hirudinea, Lumbricus, etc. are the exonephridial examples. On the other hand, the enteronephridia opens to the excretory canal or alimentary canal. They do not open directly into the exterior because they lack nephridiophores. The septal and pharyngeal nephridia of Pheretima postuma are the examples of enteronephridia. Chloragogen cells. These are star-shaped and also called as Y cells. These cells are derived from the inner coelomic epithelium with excretory functions. These cells are found in earthworms. They are yellowish in color due to the presence of yellow granules called chloragosomes. The cells stores glycogen and neutralize toxins and are present in the coelomic fluid of some annelids. These cells function as liver in annelids. They store and transport nitrogenous waste, basically urea. This is the diagram showing the chloragogen cells with a central nucleus. We have come across the different types of nephridia in previous slides, but there are certain combinations of coelomoducts with nephridia. These types of structures are called Nephromixia. We can say this is a combination of ectoderm with mesoderm. These act as compound segmental organs. This serves for both excretion and exit of gametes. Based on the combination, the degree of combination, the Nephromixia are of following types. Protonephromyxium is a combination of coelomoduct and protonephridium. When a coelomoduct is joined with the protonephridium and they share a common duct, the structure is called protonephromixium. It conveys both reproductive and excretory product to the exterior. Protonephromixa occur in Phyllodoci. The Metanephromixa It is a combination of coelomoduct and metanephridium. These types of Nephromixia are found in Hesione. The third type of Nephromixia, the Myxonephromyxium, is a combination of coelomoduct and nephridium. The coelomoduct and nephridium are intimately fused to form a single organ. Its funnel is formed by coelomoduct and its duct is formed by nephridium. Myxonephridia occur in Arenicola where they are restricted to the 6 segments of the second tegma of the divided body. Each organ has a freely funnel as an internal opening. A rich blood supply is there. The gonadal tissue are in close proximity. The fourth types, the ciliated organs. Actually, these are devoid of nephridium, but the nephridium part, though absent, the coelomoduct also reduced to a ciliated organ. They are attached to the dorsolateral longitudinal muscles and opens externally in case of Neris. Till now we have discussed about the nephridial types, the structure of nephridia, different combinations of nephridia. Now let us have a look into the process of excretion, the physiology of excretion, which is divided into 4 parts: the uptake of materials or filtration from the blood, tubular reabsorption, tubular secretion, and the expulsion from the body. Excretion in earthworms. The body fluid of earthworm is rich in nitrogenous waste that enters the nephridium through nephrostome and passes into nephridial tubuli. In earthworm, nephridia are of 3 types according to their position in the body. Septal nephridia, which are found on both sides of intersegmental septum behind the 15th segment. The pharyngeal nephridia lie 1 pair in each of the 4th, 5th and 6th segment. The integumentary nephridia are found from the 7th segment to the last segment. The septal nephridia discharges the waste products through canal and ducts into the lumen of the intestine. So they are enteronephric. The pharyngeal nephridia discharges the waste into the mouth and buccal cavity. So they are called enteronephric nephridia. The third type of nephridia, integumentary nephridia, discharges the nitrogenous metabolic waste products directly outside from the body through nephridiopores. So they are called exonephric nephridia. As a whole, earthworm excrete about 40% urea, about 20% ammonia, and about 40% amino acids and other nitrogenous wastes. Earthworms are mainly ureotelic. Let us have a look into the physiology of excretion in oligocytes. The nephridia, as we know, that consists of nephrostome, a tubule, a bladder and an opening to the exterior called Nephridia-III. The nephrostome is a funnel-like structure which filters the unwanted materials from the blood, which includes protein, urea, ammonium ion, sodium ion, potassium ion, chloride ion, some water. The first of the tubule, the narrow tube cells, secretes additionally some uric acid, urea, ammonium ion, and salts. The cells of the middle tube absorbs salts. The wider tube cells absorbs protein, potassium ion, sodium ion, chloride ion, some water, and finally the bladder receives the urine which is expelled out from the body. This expelled urine is composed of ammonia, urea, uric acid, water, and some sorts of ions. In the concluding part, let us make a conclusion that the annelids have a system for tubular reabsorption by a capillary network before excretion. Earthworms excrete urea as main excreta, but they are less preferably ureotelic than other terrestrial animals. Though urea is present in the urine of earthworms, ammonia remains an important excretory product. The level of urea depends on the physical conditions of the worm and the environment of its habitat. The slide shows the list of books which are followed during the preparation of this presentation. So thank you viewers. If you like the video, then you can subscribe it, and if you have any queries, please don't hesitate to email me at upa_sanjib@rediffmail.com. Thank you.